All right, can I see the next slide, please?
Yeah.
Okay, so our next, I don't have the slide. Thanks, Myron. All right, so our next speaker is Dr. Al De Leon. I can't see it yet. Okay, so he had a BS Kimeng at UP Diliman, and he also possessed a PhD in macromolecular science and engineering from Case Western Reserve University in Ohio. Currently, he is a staff scientist at Thermo Fisher Scientific at Bedford in Massachusetts in the United States. He formerly held a teaching assistantship at the Department of Chemistry at the University of Houston, and he was also a former instructor at the Department of Chemical Engineering at UP Diliman. Let us all welcome Dr. Al De Leon in his talk on stabilizing echogenic nanobubbles by nature-inspired elastic contrast shell design. Sir Al, the floor is yours.
Thank you so much, Lawrence, for introduction. I hope everyone can hear me well. Let me share my screen. Okay. So it's my pleasure to talk about how we try to stabilize echogenic nanobubbles by nature-inspired elastic contrast shell design. Again, my name is Al De Leon, and this was the work I did back at Case Western Reserve University. So when we talk about ultrasound imaging, I'm sure the first thing that comes to your mind is what we use for pregnancy. But ultrasound imaging is much more than that. It's also a diagnostic medical procedure that uses high frequency sound waves to produce dynamic visual images of organs, tissues, blood flow inside the body. So there are a lot of different types of ultrasound imaging. The most popular is the brightness mode or the B mode. But unfortunately, it has a very poor soft tissue contrast. So here are three different ultrasound images. And if you're not a trained radiologist, it would be very hard to distinguish, or it's very hard to know what organs these are. These are actually liver, colon, and also prostate gland. Again, you can't tell anything because it's all just gray images. Currently, there's a new technique called the contrast imaging or harmonic imaging, which is basically just ultrasound imaging with the help of an ultrasound contrast agent. So looking at here, B mode of the liver, if we inject the ultrasound contrast agent and do the harmonic imaging, we can see that we have more details. We can do the same for the colon and also for the prostate gland. Interestingly, these bright spots, they're actually tumors. These are completely invisible in B mode, but it's very bright in the harmonic images. The commercially available ultrasound contrast agents are mostly microbubbles. So they respond differently under ultrasound as compared to soft tissues. So the microbubbles have expanded the application of ultrasound imaging. So now ultrasound can also be used to target specific cellular markers of diseases, provide dynamic blood flow estimation, and so on. But these microbubbles cannot extravasate, so they remain in the blood vessel. The signal that we also get from microbubbles can only last for 5 minutes. That means microbubbles have very limited molecular imaging applications. So what we did in the lab is to shrink these microbubbles into nanobubbles so that they can easily go out of the veins. And also we tried to stabilize it by extending the signal that we get from it for more than 5 minutes. If you look at literature, there's a lot of ways to stabilize nanobubbles. If you want to improve stability, you have to decrease this coalescence probability. And this can be achieved by using high lipid or surfactant concentration, using high molecular weight of polyethylene glycol, and high polyethylene glycol fraction, as reported by this literature. You can also use phospholipid surfactant instead of small molecule surfactant. Here, the DVPC, or the phospholipids surfactant, the bubble that's made from it can last for at least 71 minutes. In contrast, the bubble that's stabilized by the small molecule can only last for about 15 minutes. Among the phospholipids, the one with 22 carbon chain shows the longest in vivo half-life. So we can combine all this knowledge to develop our stable nanobubbles, but we can extend that stability by gaining inspiration from nature. So there's a number of natural systems that can withstand stress and mechanical deformations. Your blood vessels, your skin, your cell membrane. And what's common among these three is that they have unique interfacial structure that's composed of a stiff layer and a flexible layer. So this bilayer architecture with elastic contrast has been shown to effectively deflect cracks, redistribute stresses, and also dissipate excess energy. For our nanobubble formulation, we incorporated a membrane stiffener and also a membrane softener. The membrane stiffener enables folding when you compress a monolayer of phospholipid at the air-water interface. And this cannot be done if you don't have any membrane stiffener. On the other hand, membrane softener has been used to transform your liposomes into ultra-deformable liposomes, capable of squeezing through narrow openings without disruption of its vesicular structure. So we wanted to investigate how the membrane stiffener and also the membrane softener affects the nanobubble stability. So we performed an experiment wherein we have 3 different formulations of nanobubble encapsulating a fluorinated gas that is composed of just the phospholipid and the membrane softener. So we call it the flexible nanobubble. We also have phospholipid with membrane stiffener, and we call it the stiff nanobubble. And intermediate containing both, and later on you'll see that this is the ultrastable nanobubble. To make the nanobubbles, we first prepare the phospholipid solution containing the membrane softener or the membrane stiffener, or both. We then mechanically agitate it in the presence of the fluorinated gas, and then separate the nanobubbles from foam and microbubbles by differential centrifugation. And then we do our in vivo and in vitro experiments. After we isolate the nanobubbles, we analyze the size and its concentration. And to do this, we used a resonance mass measurement, wherein we let each nanobubble flow through a cantilever that is oscillating and look at the shift in the frequency. From this instrument, we can get the concentration and the diameter. Here is a representative data that we can get from this. And as you can see, there's no particle or there's no nanobubble that is bigger than 1,000 nanometers. We also learned from this that the diameter and the concentration did not vary significantly regardless of the formulation. We now look at the behavior of single nanobubbles under ultrasound. And here we can see that the degree of oscillation caused by the nanobubble depends on the stiffness of the shell. We can also get the Fourier transform of this oscillation. And here we can see that aside from the fundamental oscillation, we also have subharmonic, harmonic, and ultraharmonic, which is very important because our cells only oscillate in the fundamental frequency. So we can easily just remove the fundamental fraction and focus on the subharmonic or the ultraharmonic. We then analyze the ultrasound image that we can get from the nanobubble solution. And to do this, we place the nanobubble solution in a agarose channel that is placed on top of an ultrasound transducer. We vary the transmit frequency, and we learned that as you increase the transmit frequency, we improve the resolution of the ultrasound image that we get. We can also analyze how bright the signal and call it the enhancement. And here you can see that the flexible nanobubble shows the greatest enhancement among all the formulations. We continue to expose this nanobubble solution to ultrasound and look at stability under constant insonation. To do this, we use an agarose with a very narrow channel because we don't want any nanobubble diffusing in and out of solution, or in or out of the plane of the visible plane. We also vary the intensity of the pressure of the ultrasound that we use. Here you can see the images before the ultrasound pulses. So you can see that the brightness increases as we increase the pressure. After 500 ultrasound pulses, we see that there's not very much different in terms of brightness. We can say the same for the flexible and the stiff nanobubble. But if we look at the in vitro half-life, we can see that the ultrastable nanobubble has the longest. So we are thinking that this, again, is due to the fact that we have both stiff layer and flexible layer in the formulation. The membrane softener introduces elastic and compliant defects in the shell that absorbs excess energy during compression. And the membrane stiffener forms a vitrified ad layer that provides stiffness, limits strain during expansion, and functions as an additional layer resisting gas diffusion. So after learning all those in vitro, we are now ready to study it in vivo. So we injected this nanobubbles into the mouse and imaged the kidney and the liver. But as a baseline, we also imaged a commercially available microbubble known as Lumason. Here you can see in the B mode where the kidney and the liver, before injecting Lumason, we don't see any signal in the harmonic images. But 1 minute after injection, we see it lightens up, but the signal disappears within 5 minutes. The flexible nanobubble shows increase in signal after 1 minute, but that signal remains until 15 minutes. The stiff nanobubble shows the same, but the signal only lasts for about 5 minutes. And lastly, the ultrastable nanobubble shows signal all the way to 20 minutes. So again, we are comparing 1 minute from the commercially available microbubble and the 20 minutes, at least 20 minutes for the ultrastable nanobubble. We can analyze the time intensity curve, and we can see that Lumason has a fast wash-in, so it gets bright really fast, but starts to decay as soon as it reaches maximum enhancement. Nanobubbles behave differently because they remain at its maximum for some time before decaying. So Lumason follows the typical log normal behavior, while the nanobubbles follow the sigmoidal behavior. And we can fit these equations into the data and extract more parameters. We can see here that the in vivo max enhancement for the flexible, ultrastable, and Lumason are not significantly different. But the in vivo washout half-life for the ultrastable nanobubble is 12 times longer than the half-life of Lumason. And also the decay onset happens 12.5 minutes after injection as compared to 0.64 minutes for Lumason. That is about 20 times longer. We then go to imaging mouse flank tumor. And for comparison, we also image the kidney. So here you can see that Lumason, again, gets really bright. The kidney gets really bright after 1 minute, while the tumor remains really dark. And the signal disappears within 5 minutes. We can again plot the time intensity curve, and we see that, again, it goes up and then it goes down really fast. The ultrastable nanobubble for the kidney shows the sigmoidal curve. It remains at its maximum for about 15 minutes before it starts decreasing. But if we look at the signal from the tumor, we can see that the contrast dynamics are very different. Maximum enhancement for the ultrastable nanobubble is 2 times higher than that of Lumason. The in vivo half-life is 12.5 times longer. And also the decay onset happens 11 times longer. So this is the movie comparing the ultrastable nanobubble versus a different commercially available microbubble in a flank tumor at the back of a mouse. So we can see here that there's higher enhancement. The signal is brighter for the ultrastable nanobubble. And also the nanobubble highlights the entire tumor as compared to the micromarker that's only highlighting the periphery of the tumor. After 5 minutes of imaging, we also did 3D imaging. We moved the transducer across the tumor, and we can see that even after 5 minutes, we have signal from ultrastable nanobubble. We estimated the amount of conscious agent still active in the tumor to be 91.2% for the ultrastable nanobubble after 5 minutes. But for the microbubble, it's only about 20%. So because of this, we now have a flexible platform for various applications. So this ultrastable nanobubble formulation can now be used to study different effects, different like anesthesia effect on the ultrasound parameters for nanobubbles. We can also use it to image orthotopic prostate cancer with PSMA targeted nanobubble, and also monitor in real time the prostate cancer using PSMA targeted nanobubble contrast agents. So in conclusion, we developed an ultrastable nanobubbles with membrane structure that's similar to the bacterial cell envelope, to your blood vessels, and it has minimal signal decay when insulated continuously and has longer in vivo half-life and delayed onset of in vivo signal decay. It has, despite having a bilayer structure with contrasting elastic property, it still has the higher harmonic required for harmonic imaging, and the enhanced in vivo stability of ultrastable nanobubble can advance the use of ultrasound in molecular imaging, theranostic, and also drug delivery. So with that, I would like to acknowledge our group from Case Western Reserve University with my advisor, Dr. Agata Exner, and also in collaboration with Dr. James Basilian and Dr. Michael Colliers at Ryerson University. And thank you.
Thanks so much, Doc Al. So any question from the audience? Please raise your hand. Yes, Brian, go ahead.
What's up, Brian?
Okay, hi, Sir Al.
Hello.
Nice talk. I just have a question on, because you showed earlier a plot wherein you have the size of the diameter of the bubble is not changing irrespective of the formulation. Do you have any insights on what limits that size?
So the way we manufacture the nanobubble is that we have the phospholipid solution and then we shake it vigorously in the presence of the fluorinated gas. So with that procedure, we actually produce foam, microbubbles, and nanobubbles. It's just that we isolate the nanobubbles via centrifugation. That's why we have the same sizes of nanobubbles.
Okay. So is there any physical mechanism or any interfacial mechanism you think that's trying to at least set the size within that range, or it's just
I think
because of the experiment?
Yeah, I think it's mostly on where we collect the nanobubbles after the centrifugation.
Okay.
After the centrifugation.
I see. Maybe a second question. You mentioned about earlier about the presence of defects in, I think, in the flexible layer. Is there a rule of thumb, like how many defects should be available or should be present so that we can have the flexibility, to be beneficial to the application that you're looking at?
I'm not sure if there's a way to actually count the number of defects, but the membrane software that we use has been established to introduce those defects. I don't know if there's like a paper that quantified the amount of defects, because mostly after they introduce the membrane softener, they just look at what happened to the membrane flexibility.
Okay.
But probably with like molecular dynamics simulations, maybe you can do that.
Okay. All right. Thank you.
You're welcome.
Thanks so much, Brian. Any other questions? I think I saw a hand earlier, but please try. I think I can ask the question, Sir Al. So,
Sure.
what's the commercialization stage of? Are you into commercialization, or what's the status of this improvement, advances in harmonic imaging
Mhm.
that you did at Case Western, and how is it related actually to your present work in Bedford?
Oh, so it's not related to my current position in Bedford. So I left Case last year, but I think they're still pursuing the nanobubbles. Right now, they're doing studies in bigger mammals. So we're done with mouse. So they're doing rabbits now. And I think the next step would be to go to dogs and then to bigger mammals. Um, in terms of commercialization, they need to get like an FDA approval first before they can do like human testing. But yeah, I think in terms of stage, they are doing animals bigger than mouse.
All right. Yep. So, any other questions? We still have about 2 minutes actually.
I have a question,
Yeah,
Lawrence,
go ahead,
for
Joy.
our speaker.
Mhm.
Thanks again, Sir Al.
You're welcome.
My question: are there any other groups outside of Case Western pursuing a similar approach or a different approach to improving contrast in imaging?
Mhm. Yeah, there's a couple, but there's still a debate whether nanobubble actually exists. So we offered a lot of proofs that it exists, but there's a lot of groups that still pursue microbubbles. Um, I think there's at least like 5 groups doing nanobubbles, but with different formulation. So we are also collaborating with different groups. So we supply them our nanobubbles, and then they activate it on site and then do the study.
All right, thank you.
Okay, thanks so much, Sir Joy. Thanks so much, Sir Al. Again, join me in thanking Sir Al all the way from Massachusetts tonight, this morning. Hope you're keeping safe there, Sir Al, and yeah,
Thank
staying
you.
healthy as well. I'll now turn over to Sir Joy.
Thanks, everyone.